Targeted protein degradation, or TPD, is a way to knock down proteins directly rather than knocking down the levels of the messenger RNAs that go on to make the proteins. The way that this is done is by taking advantage of the cell's own machinery for breaking down proteins, the Ubiquitin-Proteasome System. If we have a way to direct specific proteins to the proteasome, then we can degrade them on demand, which is great if we want to instantaneously get rid of the protein and not have to worry about waiting for the results of the gene knockdown to kick in. And it's great in that you can actually use small molecules in order to do this rather than having to introduce nucleic acids, which can be hard to get into cells. You can titrate the levels so you can control the amount of protein that's present. You can reverse things by just getting rid of the molecule that you were using to induce the degradation. Techniques that are often used are PROTACs, molecular glues, and conditional degron tags. PROTACs and molecular glues, these, if you're lucky, can be used on endogenous, naturally occurring proteins. These have promise for things like actual use as pharmaceutical drugs, especially for proteins that are hard to target in other ways, such as if you can't find an inhibitor of them because the protein's too similar to other proteins, or if there aren't any good kind of druggable sites. The conditional degron tag, this is great for an experimental setup because you can do this to kind of any protein, but you have to do it on a kind of added version of a protein or an edited version of the protein that was already there. But this allows a powerful experimental tool. How these tools all work is by directing your protein to be ubiquitinated in order to be degraded. To add that ubiquitin, you need the ubiquitin ligase. If we can target that ubiquitin ligase to a specific protein, we can get that protein tagged and get that protein degraded. The way that you can do this is by using a molecule called a PROTAC, so a proteolysis targeting chimera. Chimera, you've got a kind of mismatch of two parts. One part binds your protein, one part is the ubiquitin ligase that adds the ubiquitin. If you have a molecule that'll bind your protein, you can find these in various ways. There's like small molecule things, like a pharmaceutical drug type of molecule. Now what you do is you add a linker to the ubiquitin ligase. This part binds to your protein, this part adds the ubiquitin tag, voila, your protein gets degraded. You can also have a molecule called a molecular glue. Instead of having chimera where the two parts are attached with a linker, the parts are separate. When the small molecule binds to your protein, it's going to make it so that the ubiquitin ligase now is going to stick and add the ubiquitin and then get it degraded. Then you have the conditional degron tags, where here you introduce a tag onto the end of your molecule that will bind to specific molecular glue, such as the auxin-inducible tag, where you can add auxin. And then basically what happens is it'll serve as the bridge to the ubiquitin ligase, add that tag, and then you get your protein degraded.